That looks great. That looks great. And thank you everybody online for helping out and telling us. Oh, brilliant. So Helen, with no more delays, it's over to you. We're really looking forward to hearing from you. Go for it.
Okay, so I'm happy to be here. I'm going to talk to you about building community resources for structural biology. And it's basically, for some of you, you probably already know a lot of what I'm about to say, but I think it's important to look at the whole history of what happened in structural biology to understand how the PDB got to be what some people say is special. When we talk about the data, they'll say, oh, the PDB is special. It's not really the PDB that's special. What's special is the way in which the community built the PDB. And that's what I'm going to emphasize in this talk. Okay? So the next slide. This is very important. J.D. Bernal was a very key influence in the culture of structural biology. He's a crystallographer, but he was very interested in the social function of science. And this particular book covers the organization of research to science and the social role. And there's a lot of things covered here, but his influence on the community was very, very great in the way the community functioned. The pioneers of structural biology, probably all of you know, Dorothy Hodgkin was the person who did the first diffraction photograph of a protein crystal. This was done in 1935. She was working with Bernal. And of course, later on, went on to win a Nobel Prize for her work, actually not in proteins, but in small molecules. John Kendrew did the first crystal structure of protein, myoglobin, and Max Perutz shortly thereafter of hemoglobin. And these were pioneering structures that people, that influenced many people, including myself, who was growing up at that time and knew a little bit about what was going on, and it influenced me very greatly. What was an extremely exciting field. The very first structures were myoglobin, and then hemoglobin, and then lysozyme by Phillips. And you'll note that these three structures are all done in the UK. Americans were very jealous. I know that. I was a student at the time that this work was coming out. And so when ribonuclease was determined by the group in Buffalo, led by Cartha, and Yale, led by Fred Richards, it was met to, we had a meeting in Atlanta, Georgia, American Crystallographic Association, and it was a packed house to hear about this one structure, because finally the Americans had something to show for all the work that was going on. So those were the first structures in the 60s. At the same time, there was the intense interest in protein folding, and there was the work by Chris Amundsen, in which he was able to show that you could have reversible folding and unfolding and folding again in vitro. And that led to the idea that sequence had the structure in it somehow. There was a code for the structure. We didn't know what that code was. At the same time, there was the pioneering work of Cyrus Leventhal, made people in silico. Cyrus at the time, this picture is taken in Michigan, but he spent a good part of his career in MIT, and he was very involved in computational work at what was called Project MAC. and he really wanted to understand sequence structure and protein folding. And many of you know about the Leventhal paradox about if you have to, if there are so many degrees of freedom in the bonds that are being changed in order to fold a protein, how come it happens so fast in real life? And he was fascinated by that. So those were the sort of in vitro and in silico motivations for trying to understand protein folding. So while this work was going on in the 60s, and some of you know that the 60s was a very tumultuous time in the world, and maybe not as tumultuous as now, but pretty close. And the way in which we operated as scientists, some of us, we were in a very activist mode. And we didn't follow the rules that some people follow now. We just did things in a very activist way. So in the next slide, I show you what happened in order to create the Protein Data Bank. There were three of us. Edgar Meyer, who was the person who was really interested in visualization of proteins. Gerson Cohn was Edgar had been at MIT, and then he had gone to Texas A&M, but he spent his summers at Brookhaven. So Edgar, Gerson, and myself felt very strongly. Gerson was a postdoc working for David Davies at NIH. I was a freshly minted PhD student. So we were all in the early parts of our career, and we really wanted a protein data bank. We were very energized by that idea. So we had lots of meetings, and we corresponded by snail mail, no email. At the same, but we were not protein crystallographers. There were separate discussions among protein crystallographers about the need for having a protein data bank. And in that case, it was like purely practical because people wanted to see the coordinates. The only way at that time you could do that was to ship a box of punched cards, and that was very, very impractical. So we were very active. We wrote all kinds of letters to each other and to other people. And then there was a meeting in the February of 1971 in which we it was a meeting saying we need to have a PDB. And we wrote a petition. We had absolutely no idea who was going to read this petition. You'll see some interesting names on there on that petition. We sent it to the American Crystallographic Association because that's all we knew. We didn't know what else to do. And they took a very supportive role and tried to help us figure out how to do this. Next slide.
Slide number, please.
6.
Yeah, okay. Yeah, I'm in 6.
So that first meeting, the big meeting that we had with the petition was in February of 1971. In June of 71, there was a Cold Spring Harbor meeting that in protein crystallography, and it was an amazingly exciting meeting, invitation only. But again, us being the way we were, we just went. We crashed the meeting, and at the end of one of the sessions, we met Walter Hamilton in the bar at Cold Spring Harbor. Those of you who've ever gone to Cold Spring Harbor know that after the talks, people hang out there, and actually good science can begin that way. We went to see Walter, who was a small molecule crystallographer, but interested in biology. And we said, we need a protein data bank. And he said, okay, you'll have it at B&L. And he then flew over to England to talk to Arthur Kennard because he knew instinctively that if we didn't get international cooperation, it wasn't going to happen. Edgar set up the technology at Brookhaven. I was one of the first annotators. And then we had a very novel computer infrastructure, which if you would like to know about it, I'll be very happy to explain it because it was, it's what funded the PDB actually. So in October of 71, there was an announcement in Nature New Biology that there would be a PDB. You'll note that we got the idea to make it happen in 71 in June, and by October it was set up. Next slide, 7. So the PDB was set up at Brookhaven. It was a very small operation. We had to beg people to submit their data because there was no requirement, and it was still, if you think about it, a bunch of kids doing this work without really any, we had minimal funding to do it. So finally, as the PDB was growing, within the next 10 years, people like Dick Dickerson and Fred Richards became very active, wrote letters, published articles, had a petition saying that we needed to have mandatory deposition of structures in the PDB. And when that happened, we then, there were committees set up in various places. The most important one was set up by the IUCR. And it was to set up the standards for what do you mean by mandatory deposition? What data should go into the PDB? And how should it go in? And it had a lot of leading figures of protein crystallography on that committee. And after 7 years, they finally agreed on what had to happen. Next slide. And in 1989, the deposition guidelines were set up by all of these committees that I show on the left side of the slide. And that was really, of course, not surprisingly, when you say that it's mandatory, and we got the journals to agree, and it got to be that you could not publish a structure unless it was deposited in the PDB. And having that as and then the funding agencies aligned in that way. Not surprisingly, people complied, and I think there's one of the funding agencies in America said if you did not deposit in the PDB, you would lose your grant. And only one person lost this grant because people understood how important this was. Next slide.
Yes.
Okay, so that's slide number 9. So, okay, so
Yes.
now we have the PDB and we have the old format, which is based on the 80-column punch card format. And many of us felt pretty strongly that this format was not going to work. And that it wasn't going to work if we were going to do really computational, make databases and things like that. And some of us were involved in making, myself especially, were involved in making databases, and we knew that this was not going to work. So we formed a committee called the Macromolecular Crystallographic Information Format Committee, MMCIF Committee, much hated by most crystallographers. They really hated this format. And the people involved, Paula Fitzgerald is shown in the upper picture in the middle. She was the chair. There's John Westbrook, who actually created the details of the format. Phil Bourne was involved. Sid Hall had the small molecule sieve. And Shoshana Vodak, who was really interested in validation and understood how important it was to have a good representation, were among the people involved. And so we had our first draft in 1996. We made it so that it would be extensible, meaning that if new methods came along or whatever came along, we would be able to change things in the dictionary that we formed without breaking the whole thing. So we had the format. When the PDB moved first to RCSB and then became the WWPDB, which I'll talk about in a minute, we used MMSIF, but everybody hated it in the world, we thought. And so we said the only way this is going to work is if we get the software developers on board. And so to do that, we had a couple of meetings, and the definitive meeting, the one that really worked, was one that was held at the EBI. And the idea was to get the software developers on board with this. And the first day I was moderating, and I said, if you don't like MMSIF, we'll give you whatever you want, just as long as all of you agree to do whatever it is we come to. And they understood that. And so we came up with something called a hybrid format. And the second day of the meeting, I got up and said, okay, we've agreed to the hybrid format. And I won't tell you who said, no, we didn't agree. It was somebody who was really not, he was pro PDB format, anti-CIF, but they apparently, everybody agreed that since we already had libraries and all that, it was stupid to start all over again. So finally in 2011, it became, the recommendation was to make it the master format for the PDB, and it is. And later I'll tell you just how important that was to what happened later. Next slide. So here's a simple drawing of the architecture of MMSIF. There's a dictionary definition language. There's the actual dictionary, which has all the definitions, and there are thousands of definitions in the dictionary. And then there are the actual data files, which are in MMSIF. And all of this is machine readable. We have the characteristics of having relationships among the data items. The thing is set up as a bunch of tables so that it would fit into a relational database easily. Next slide. Okay, so in 1999, there was a competition in the United States where there's a call for proposals to have for the PDB. And this was relatively new idea. The first one was, I think, in 1996, and that one Brookhaven competed and won. In 1999, they issued another one, and this time a group called the Research Collaboratory of Structural Bioinformatics, RCSB, won. And here is a picture of us at the NSF when we got the award. And you'll see a younger version of me. There's Gary Gilliland at NIST, Phil Bourne at SCSC, and this John Westbrook, Lynn Tenaik, Peter Osberger, Phoebe Fagan, and Helgi Weissig. So these are all people who were involved in that very first new RCSB PDB. Once the thing moved from Brookhaven to us, it was very clear. This opened the door for other databases and other PDBs. And indeed, there was the beginnings of at EBI, the MSD, which later became the PDBE, and there was also the PDBJ. while it was really good to have more effort on this, the danger was that we were going to break the single archive. So Haruki Nakamura, Kim Henrik, and myself got together for some reason in San Diego. I don't really know why we were there exactly, and said, no, we have to have a WWPDB. Next slide. So the WWPDB, the idea was that the data would be freely and globally available to anybody. It was really important to us that it be free and that there would be no charges, and that we also agreed that we would collaborate on data processing and annotation, ideally to have a single system for all of that. At the time, we did not, but we agreed we had to, so that no matter where you went in the world, you would always get the same data. We also agreed, and this is important when I talk to you a little bit later, we also agreed that we would compete on our website and we could offer different services and views of the data. And we felt that that was the only way to get sort of global agreement on doing all of this. And so we set up a charter. It was very formal. It was co-signed by the different organizations that we belong to. In my case, it was Rutgers. In EBI case, in PDBE case, it was EBI, and the Japanese case, I don't remember who the organization that had to sign for it, but this was a very, very formal and detailed charter that we would follow. And then we set up advisory committee that met once a year, and that we consulted, and it was a completely global committee. You'll see a picture of the advisory committee from 2013, 10 years after the inception, and you'll see Janet Thornton in the bottom row. You'll see Angela Gronenborn, a lot of luminaries in the structural biology world who were pretty strict with us about how we were going to run things. And we had those meetings every year in different places. Next slide, please. Okay, so that's the governance and how we set this all up. One of the things that we didn't have in the PDB is mandatory experimental data. So how can you actually validate a structure if you don't have the underlying primary data that you use to solve it? And there were lots of people in the community who wrote nasty letters, nice letters, all kinds of things saying you absolutely have to have experimental data as mandatory in the PDB. And finally, in 2008, we were able to require structure factors. And then shortly thereafter, chemical shifts for NMR. And that then allowed us to be able to actually validate the model against the experimental data. Again, we had to have community buy-in. Without it, this could not have happened. What happened could not have happened. Then the next slide. As you all know, the PDB began with only X-ray data because that's all we had. But as NMR began to be able to, NMR methods were being used to determine structures, we began to get structures with NMR determined, that would determine from NMR, and we needed a way to handle that. BMRB was established for NMR data, but the actual coordinates were going to go into the PDB. And so in 2007, BMRB joined the WWPDB as a member. So again, new method, we needed to get more representation of a new method. The next slide. Electron microscopy, the first structure was in 1990, the famous bacteria rhodopsin. And in 2002, EMDB was established at EBI, which was a place that you could put the maps for the electron microscopy determined 3D determined structures. And again, we needed to figure out how to collaborate, and so that we didn't break the archive. And so EMDR was established as a collaboration with EMDB and the RCSB and WACHU, okay, as another way of getting the input from the domain expert as well as different database focus groups. And then Empire was established at EBI. And then in 2021, EMDB joined the WWPDB. So again, we had to have a sort of balance between independence and cooperation so as to serve the community properly. Next slide. Okay, so the PDB, until very recently, the structures in the PDB were each solved by single methods, either X-ray, NMR, or EM. But actually, there were structures being determined using several different methods, which was integrative structure determination. And this is a slide from Andre Sali, which shows you all the different kinds of methods that can be used to determine structures, and then eventually get an actual structure, an ensemble of the optimized structures. So we didn't have any way to handle those structures in the PDB at all. And so we needed to figure out a way. Next slide. So again, we did the same thing we always do. We had a meeting, we had a task force, and this was held at EBI, and it had experimentalists from the different methods. It had computational biologists of from various methods and various places. And we said, oh, we came up with a plan for how we were going to do this. And that's when we began to figure out how to archive. We knew that we had to, and then we had to figure out ways to archive structures from that were solved using integrative methods. We came up with the idea, next slide. We came up with the idea of federating interoperating data resources. And this is the model we came up with. It's not actually the model that's being used now, but I predict it will be the model. And this was a meeting we had in 2019. And the idea is that all of the databases for these different methodologies would continue to operate the way they want to operate, collect whatever they want, but they would exchange the restraint data with the PDB. And that way they would be independent, but we would have what we need to validate the integrative structures. So that was the concept that we came up with. And in fact, there's been significant progress in establishing the Federation. This slide is slightly old, but different communities came up with different ways of standardizing the way in which they handle data. One of the most active ones was the Saxon Sands community, and Jill Truello was a very, continues to be very active in this. There's also there's work in the various kinds of cross-linking, FRET, EPR, and these groups, some of them followed a model that used SIF, some of them followed another model. Didn't really matter. What mattered is if you really standardize properly, you can exchange data. And so there's been a significant progress in that area. Next slide. Okay, and so with all that, we were able to form a data resource. This took several years, actually. The whole process took almost 10 years, but we do have a PDB IHM, which is a place to work to put integrative models. And very, very recently, within the last year, it is now possible to deposit integrative models in the PDB, and it's processed using methods that we developed for the integrative structures for validation. And this is a work in progress. It's being led by Brenda Villette at Rutgers. And there have been several papers now published about how this is being done. So again, a lot of community input to do all of this. Okay, so one of the ways in which we did everything when we needed to get validation standards, and I've shown you a little bit of this before this, is we set up these validation task forces. And the validation task forces had the experts on each method determining what it is that should be collected into the PDB, what should be validated, how they should be validated. By far the most advanced of the groups in terms of, and that's not surprising since X-ray structure determination was the earliest in structural biology, was the X-ray validation task force, which was just fabulous. That was set up by Randy Reed. There were 17 members. There's some wonderful, the upper right picture shows the rather animated conversations that people had about how to validate the structures. Jane Richardson was involved, Janet Smith was involved, Axel Brunger was involved, all led by Randy. And they came up with a way to do this. Same thing, NMR is not as far advanced in the process, but they did also come up with validation standards. EM came up with validation standards, small angle scattering, and then most recently work in the integrative hybrid methods. So in each case, we brought in people who really understood that field and had them make the decisions about what to do. Then once they make that decision or made the decisions, there had to be a refereed white paper, and then the PDB would then implement the recommendations. So you'll say, boy, that's a lot of stuff to do, but it takes a long time, but it makes it very strong, because if you have the people who are the experts saying this is how you should do it, and they spend a lot of time on it, you have a much better chance of having a good standard that people will accept. And this is work that continues because new methods come up, new ways of doing things come up. So it has to be evolving. It doesn't stop with whatever happened in 2008 or 2011 or whatever. We continue that process. Next slide. Okay, so the Protein Data Bank today, there's more than, we started, there were 7 structures. We officially decided there were 7 structures at the beginning, but I'm not sure that's really true. There are about 250,000 structures of nucleic acids, proteins, macromolecular machines. In 2025, there were more than 20,000 new depositions just in that year alone. When the RCSB took over the PDB in 1999, there were 9,000 structures total in the PDB. Now there's 20,000 a year coming in. As you all know, the users include industrial, academic scientists, lots of disciplines, teachers, students, the general public. There are 60 million, and I think this is an underestimate now, coordinate files downloaded every day. So PDB is very heavily used. and it's used by more than 400 other biological databases, because one of the things that's really important is that data are reused. You don't want to have a database that nobody even looks at. Next slide. So what has DataShare enabled? For the depositors, at the beginning, it was just simply prevention of data loss, because it often happened that people, graduate student does a structure, leaves the lab, and people might not know what happened to the data. And we did have one amusing thing happen from a very famous crystallographer in a total panic because she got a new computer, and she said, my data is in the dumpster in the back of the building. And so did we have her data, her original data? And we said yes, and we could help her. Of course, it allows for efficient structure determination by molecular replacement, which is the most popular method. And then map fitting becomes easier because you have other examples of similar structures. And it has allowed us to create new validation methods. What is data sharing enabled? Well, there's a whole new field called structural bioinformatics, which allows for classification, comparisons, and analysis. New knowledge bases. There are lots of specialized databases, subsets of the PDB where people add more information, and that's great. And that's so specialists can go to their particular database, which is all using the data from the PDB. You all know about drug discovery and how key the PDB has been to drug discovery. And then protein design, David Baker, and structure prediction, John Jumper. And these are all things that we could have hoped back in the 60s that this would happen, but it actually did happen. And I think it's just amazing that all of this could have been. When I asked the people at AlphaFold, DeepMind, what was important, they said the fact that the data was highly curated and very reliable, and also that it was in the machine readable form, and mainly MMSIF. And so the people who did the work at AlphaFold were pretty young and didn't have the loyalties to the PDB format that the older folks did. And so they used MMSIF, and it worked very, very well. So what does it take to make a good database? You have to follow the science at all times. You have to follow the technology. So all of you know, I think most of you know that the methodologies, the science gets more and more complicated, the structures get more and more complicated and more interesting. And the technology, both for determining the structures and handling the data, has evolved enormously. But really, another key to all of this is the community. And I hope I've shown so far how important community really is to how the PDB operates. And so I'm going to talk to you a little bit now about, more formally, about community and how that works. So the community input is essential. So what do we mean by community? First of all, the data producers. Without the data and without the structures, there's no PDB. The software developers, the people who are constantly improving the software to allow you to determine structures. The database experts who are constantly looking at newer and better ways to handle the data. The journals are really important because you have to publish your structures. And if we can get the journals on board, which we have, maybe too much, that is really, really important. And then the funding agencies. who have an influence, obviously, on what we do. So how do we get the input? We have to have workshops to focus community discussion on the data dictionary development. The more people involved, the better. Next slide. So here are the scientists, no matter what, without the scientists and without the structures, there's no PDB. And so here are some early papers of protein structures. And then the beautiful picture by David Gutzel of the kinds of structures that are in the PDB, which all of you know is very, very diverse. Next slide. The biocurators, these people review the details of every single structure, and this is they're biocurators in all the centers. Almost all have PhDs in structural biology. They're globally distributed in America, Europe, and Asia. They have regular meetings in person to discuss issues, as well as Zoom meetings regularly to figure out what they're doing and what's better ways of doing things. When they leave the PDB, they get good jobs in academia and industry. So these are pretty high level people who are very devoted to what they do. And everyone is involved in outreach. They write papers, they go to professional society meetings. So it's not somebody in the corner editing data. That just doesn't happen. So next slide. Then there are the software developers that create the tools for the biocuration. And there are tools for deposition and validation. The tasks are divided among the sites, and they also have regular meetings and summits. So this is a meeting that was held at EBI in 2017. And of course, we have pictures of everything. We document the whole thing. Next slide. This was probably the most ambitious meeting and the most important meeting we ever had. It was in 2007, and we somehow managed to get all of the teams from around the world to come to New Jersey. We went to a conference center, and we tried to figure out how we could have a single tool to handle all the data at deposition, because until then, because we were using different tools, the data had to be exchanged every night among the centers to double check that everybody would get the same answer. And that's really a waste. And we knew that, and that came from merging different groups. We had to have a single plan. So we got together for a couple of days. And here on the lower, so the top picture shows the whole gang. The lower left shows the people at the time who were leading the efforts, and you'll see Harald Kleivik and Kim Henrik have a crown on them for some reason, and figuring out what's important and using all these sticky notes and doing things that most academics are not used to doing, but it really worked very well. And then everyone then took some of the suggestions, and there were roundtable discussions until we came up with some kind of plan for how to do that. And that plan was then formally executed. It took a long time to get it right, but we were able to get a common tool, which is now called OneDep. As I showed you before, we had the external task forces for validation, which continue to meet to this day. Next slide. And then we have committees to provide advice and consultation. So there are advisory committees for each data center, RCSB, PDBE, PDBJ, and now BMRB and EMDB. They meet yearly to discuss policies and ensure that the archive meets high standards and continues to be open, which is super important, especially these days in America. Next slide. So how can other communities follow the example, the PDB example? So that this whole issue about what's special, I think any group can do it. What's the problem? You have to define what's the problem you want to solve. And it turned out with the PDB, the problem we wanted to solve back from the beginning was the sequence structure relationship. So the fact that the PDB file had both the coordinates and the sequence in the same file turned out to be really important. You have to reach out to other people who have similar interests. You have to figure out what data are needed to solve the problem. And then you have to create a data dictionary of well-defined terms that's completely machine readable. And then you can create a database. And I think this is doable. And having done it, I will tell you, it's also a lot of fun. It takes a long time. You have to live long enough. But I think it could take a lot less time if we follow the example of what we did at the PDB. Next slide. So what are the principles? One of the things I wanted to learn when I retired from the PDB are there any principles that guided what we did? I know what we did. Well, it turns out I got to talking to people and they said, oh yeah, that has to do with a theory by Eleanor Ostrom, who won a Nobel Prize in economics in 2009. And her idea, what she said is top-down enforcement simply doesn't work. And she believed in bottom-up, meaning
you'll need to unlock your iPhone first.
So my iPhone just talked to me for some reason. So she said that you had to devise clear rules, what actions are not allowed, has to be based on local conditions and knowledge, adaptable to changing circumstances, and organized monitoring and enforcement, which is exactly what the PDB did. But we didn't know we were doing that, but that's what we did. Top-down does not work. It's hierarchical, and the organizational leadership defines the goals and makes decisions. Well, if you don't have the people doing the work, helping to make the decisions, it's simply not going to work, okay? So I didn't know that when we were doing it. I learned it after the fact. So in the last few years, I've joined a group called the Stakeholder Alliance. And I wanted to learn more about this and how this works. This is an international group. It meets weekly by Zoom. It's led by Joel Gershenfeld, who is a sociologist at Brandeis, and he does things like labor union negotiations and things like that. But the members of the group are scientists, physicians, businessmen, psychologists, sociologists of all ages and stages. We have students in the group, and we have very, very senior people. And our goal is to understand how collaborations and consortia work. Next slide. So the characteristics of a consortia, which is what the PDB is, it's both independent and interdependent. You need both. You have to have common and competing interests. You have to have sufficient alignment for action. It's a dynamic and continuous, there are dynamic and continuous processes, and addressing together what cannot be accomplished separately. Well, you know, that's what the PDB did. We didn't know that's what we were doing, but that's in fact what we did. And why consortia? Because most global challenges and many local challenges exceed the capacity of one organization or institutional actor. Next slide. So we began, we have all these meetings. We wrote one paper for the Stanford Social Innovation in 2022. And we came up with this idea of, instead of bottom up, we came up with the idea of lateral alignment. And what we mean by lateral alignment is you have multiple groups who are doing all kinds of different things, but they have a couple of things in common. And those are the things that they collaborate on. And then you have other things that you compete on. And it has to be very fluid. And so we talked about what you needed to have what we called a minimal viable consortium, what will work. And it absolutely has to be fluid. Next slide. We came up with a model for alignment that involves having shared interests and a shared vision. There has to also be separate interests. You have to have a charter, and you have to have continuous change in order for this to work. So what I did was say, how does the PDB align with the stakeholder alignment model? So you have to specify your stakeholders. And in our case, the stakeholders evolved with the science. You have to identify the interests, which is high quality and open data. That's what we really cared about. We had to build a shared vision. And the shared vision was to help understand sequence structure function relationships. There had to be active and adaptive governance, which is why we have, I think we're up to the 3rd charter for the WWPDB, and regular meetings. Internal alignment means that each individual group, like the RCSB or the PDBE or PDBJ, has to have clear governance. So they have to operate independently well in order to be able to share what needs to be shared. And we have to achieve alignment for action, and our action was, I think, that we have a pretty successful deposition and distribution system. So this was an attempt. I wanted to understand what we had done after the fact, because in order to, once we understand it, then maybe other groups can take those lessons and use them to form their own consortia. So here's the next slide. I'm going to have a few slides just telling you who did what. The PDB ecosystem for curation and dissemination, you'll see there all these. The WWPDB is a sort of overall organization. There's an RCSB, PDBJ, PDBE, EMDB, and BMRB. And then we cannot forget the original PDB at Brookhaven from 1971 to 1999. The users are many, and the funding is not the way it should be. There are agencies from, well, I guess I should say we're lucky we're funded at all, because it's agencies from around the world with regular renewals, and at any point, funding can be lost. Now, one of the advantages of having the WWPDB is that when there is a crisis, funding crisis, or a natural disaster such as Fukushima, the other groups can jump in and process the data until the crisis is over. And so we do have regularly supported one another in our efforts to get money. And I don't know how many agencies there are involved in funding the overall international effort. Here's a picture of all kinds of people who've been involved in the PDB. On the upper left is Tom Ketzel, who ran it after Walter Hamilton's death for many years. And then Joel Sussman came in 1993 and took over at Brookhaven. Here's a picture on the upper right of the group. We had a PDB 40 meeting at Cold Spring Harbor where we brought all the teams together from the beginning. You've seen the pictures of the one-dep group trying to figure out how to do a one-dep. And then the picture in the middle shows one of many advisories. And then an important picture on the lower left is of Olga Kennard and myself and Janet Thornton and Shoshana Vodak at a PDB anniversary meeting in in Jerusalem. I think it was in 1990. I think it was 1996. And Olga is very important in all of this because she set up the Cambridge Crystallographic Database and, stuck with it and is still around. Unfortunately, Olga passed away a couple of years ago, but she was very inspirational in figuring out how to make a database. Next slide. A special tribute to Walter Hamilton, who was the person who said, yep, we could do it. And this was at Cold Spring Harbor. Edgar Meyer, who did the early technology for the PDB. Fred Richards, who was an unlikely activist for saying that there had to be mandatory deposition. Michael Rossman, who was always deeply involved in making sure that the PDB was doing the right thing. John Woolley, who was somebody from the NSF, who was a big proponent of an international PDB. He passed away 10 years ago. And then Marvin Kastman, who was at NIH, who was the person in 1982 who told us that, I'll say it, he said, I'm not going to fund any AIDS research in structural biology unless those data are mandatory. He was not supposed to say that to us, but he did, and that was really important. And next slide, just the book that we wrote from the study group that I belong to, and it's called Consortia Century, and it talks about how you can align in order to do collaborative work, and it just came out last year. So with that, I would like to say thank you for inviting me, and I would entertain any questions.
Thank you so much, Helen. That was absolutely amazing. So do people have any questions? They can post questions. You can post questions in the chat, or you could post questions in the Q&A, and I will keep an eye on them. So I have a question, Helen, if that's okay to get us started. So
Sure.
So obviously structure determination is getting a lot more complicated because we're starting to look at more biological structures that are made up of many different pieces. What do you think the main challenges are with moving forwards into the future? What do you think are the main things we're all going to have to solve?
I can't answer for the challenges in determining the structures. The challenges in validating the structures is what is really key. And we have now a subcommittee. This is a subcommittee led by Andre Sali, trying to come up with a more holistic way of validating structures. Because right now, it's method specific, but when you're talking about integrative structures, that becomes a real problem. And one of the big issues that's going to come out is how uncertainties are defined in the different methods, and how you describe error in the different methods. And even with X-ray, in small molecule crystallography, it's pretty clear where all the errors are and how to define the errors. But as soon as you're no longer doing full matrix least squares, it becomes a big issue. And that's for a very old method, okay? And then when we start looking at the newer methods, it becomes more difficult. And so how we're going to combine the different kinds of errors in order to have a holistic way of doing validation is a huge, and the reason that's important is how are you going to believe the structures if you don't have a way of defining the errors?
Yeah, that's very interesting. So Martin is posted in the chat. He's also raised a very interesting point, of course, about dynamics, which is
Yep.
what the DRIIMB Consortium are really interested in. So Martin says, the PDB have a shared vision around sequence structure relationships. Does that need to evolve in the light of new focus on time series, dynamics,
Yes.
fragment screening?
Yes, everything has to evolve. Yes, of course. And I'm really happy that, I mean, one of the things is people would say, we really have to do something about molecular dynamics. Well, the only people who can do anything are the people who are doing the work. They have to somehow define what they want to do. And if you can articulate all of that clearly, I think something will happen. I don't know what will happen, but something will happen. But
so I am going to invite the audience to ask some questions, because the audience is being unusually shy today. So please, our community, do ask Helen some questions. So Martin, I don't know if you have anything else that you'd like to say about dynamics.
Yeah, I mean, I think it wasn't just dynamics. It was a more kind of broad philosophical point that I guess in some ways the good old days were the simple days where you went from sequence to structure, and parallels, you know, the success of AlphaFold, because it was a very well defined problem that could be solved by throwing computers at it. So I think things are now getting more complicated, and I wonder whether the glory days of answering a simple question are over.
well,
It's just being very philosophical.
No, I think, well, I like that. I think the idea of you have to define your problem that you want to answer. And if the problem now is much more complicated than what the problem was before, then that has to be defined. And I have found in my experience is if we get people together, it doesn't have to be at a formal meeting, it could be at dinner, and try to figure out what are the new problems. Can you define the problem? So the problem can't be, oh, I want a database. The problem has to be, I want to answer this question.
Yeah, I mean,
so in the early days,
it always goes back to hitchhikers. Yeah. The question is harder than the answer. And I think there were some questions in the chat, so
excellent. So I've got one from Gerland, and then Doriano's got their hand up, but I'm not sure if I'm going to be able to unmute them, so they might have to post in the chat. And then so wonderful, thanks a lot, says Gerland. Please, can you go into more detail about funding? Which countries contribute most, and do we need to worry in view of current politics?
The answer is we have to worry a lot. It's not done. So what happens is the RCSB PDB is funded by a consortium of NIH, NSF, DOE, and competes every 5 years, and has to write a very, very detailed proposal. I'm not sure now what how PDBE is funded this moment, but it's by a different funding mechanism than US. There are now joint BBC, well, you tell me the initials, but there's English American collaborations on some aspects. PDBJ is also funded independently by some government body in Japan. We're all in a different cycle, and we all have to sort of sing for our supper. And it's been my view forever that this is really, I think singing for your supper is a good idea, but there needs to be there's no cooperation among all the agencies. There's one group that is trying to do that, to advocate for cooperation and have more cooperative funding, but we really don't. we don't have what we should have for something that's so important now for doing science. So it's not easy, there is, as I said, there's one attempt. It's ongoing to have some kind of global way of doing it, but we're very far away from that.
Yeah, that does sound very worrying. I do wonder, as the world becomes more difficult and more complicated, the things that we want to achieve become more challenging. So Doriana, could you please type your question in the chat if you have a question, because I can see someone's got a hand up, but I'm unable to mute them is the problem. So is there anybody else got any questions? So are there any early career researchers who would like to ask Helen something? We're not at all scary, so we can have a lively discussion if people would like to post their questions.
Yeah, and I think the person with their hand up, if they want to try unmuting and talking, I think we have allowed it for them.
Okay, so Doriana, if you want to ask your question in person, you are able to do that. But they may.
And then there's a question in the chat, I think.
Okay, excellent, thank you. I can't see that actually.
So what are your thoughts on protein structures being submitted that have been determined by AI rather than experiments?
Oh my God. This committee that I'm on now for validation, we're talking about that. I don't even know what that means. If you're using AI to fit, to use the AI determined structure to fit part of a new data set, I think that's okay.
Yeah, I think the question might have been like things like AlphaFold.
Yeah, that's what I mean. Is so the AlphaFold determined structures are all in a database at EBI, and some of them are in a database at RCSB. Okay, but they're separated from the experimentally determined data structures, and I suspect there's going to be meetings about how to handle all of this. But that's what it is now.
Yeah, I guess it's probably important that it's labeled with where it came from and what it was.
I just saw a question of why was MMSIF hated so much? Do you want me to answer that?
Yes, please. If you're happy to, Helen.
Oh, sure. Okay, so the PDB format was a very easy format to look at with your eyes. And you would think that the format was pretty simple and it'd be, no problem to submit it. So people would submit the PDB format, and we had 27 different versions of the PDB format that have come in because people didn't really take seriously what the format was that was specified. And so that was one problem. The other problem, more serious, is that because it was based on this 80-column format, there were restrictions on the number of atoms and the number of residues, which meant that for certain structures, like the ribosome structure, you would have multiple files to describe one structure, which is really silly and not, it wasn't dealing with the fact that structures were getting bigger and bigger. And in order to use the PDB format, you had to know what each column was by looking at documentation. What MMSIF does is it's self-defining. It tells you ahead of time what you're about to see. It's linked to a dictionary, which gives you the precise definition of everything that's in that file. And you don't have to be a structural biologist to interpret the file. It is ugly to look at. It's full of dots and dashes and all kinds of things, and people hated that. But one of the things we did was we stylized, there's no format required, no column-based format required for X, Y, and Z, but we had a style that would make it look like the old PDB format. I think it's very hard for people to let go of old things that they like and treasure. And it's very hard. You have to change software in order to use a new format and things like that. In the modern day, like now, I think people are, they see the value and are happy to well, many people are happy to use it. But originally, there was really a big opposition.
Yeah, nobody likes change, but
No.
somebody somewhere does, but most people don't take well to change.
No.
Yeah, so Harry says that he really likes MMSIF. So Jazz says, thank you, Helen. I think you met Jazz before online, Helen, so you know Jazz. So Jazz asks, how long does the data curation process take for each submission, and how much of it is automated versus manual?
It's almost completely automated, but there's the driver. It's a driverless car until there's trouble, and then the curator comes in. Okay, the curator does all the sanity checks and all that. The rule in the PDB from 2000 or 1999 is from deposition to completed curation cannot be longer than 2 weeks, including all of the discussions with the author. So if the author has a problem in the structure, if there's a problem found by the curator or by the automated software, the author has to agree to the change or to fix something. And those exchanges can take time, but it can't take, it needs, on average, it's about 2 weeks. We have not increased the number of curators for a very long time, even though the number of depositions per year has gone up dramatically. The number of people who do the work has not. And the people, so the reason we need to have the PhD level people is that they have to critically look at the whole result of the curation to make sure that it makes sense and that there isn't some really silly thing wrong with the structure. So those people have their eye on it. It used to be the curation was almost completely manual. It's no longer that.
That's brilliant. Thanks. Sarah, could you check for me if there are any more questions in the chat, please? Because I can't, for some reason, I can't see the chat.
I see things flashing up.
There are a couple of questions in the chat. So one's asking about what is the standard to publish a protein structure on RSBC? So like, what are the criteria for, is it allowed in or is it blocked for being a terrible structure?
No, PDB may not, we are not the structure police. We do not turn down anything, okay? So, but we tell people how bad it really is, and it's public how bad it really is if there's a bad structure, okay? Usually these things, in the new way of doing things, the validation reports, many journals require validation reports to be submitted with the paper so that the referees can see if the validation reports look okay. So, but we do not turn anything down. But there's plenty of warnings on the structure that there are problems.
Excellent. And then there's another one. Any comments on improving validation in view of rapid deposition of cryo-EM structures? So Lothar saying, I noticed things that have been known since the 1990s are done incorrectly today. And I assume this is because of the speed with which the work is done.
No, not necessarily the speed. Are you talking about the speed of the experiment or the speed of the annotation, curation?
Well, I guess the analysis that the people creating the structures are cutting
yeah.
corners.
So that's going to be, so there's a lot of heavy duty work going on in cryo-EM trying to come up with better validation standards. Okay, for sure. So that's an ongoing issue, and people are very aware of it. But how long it'll take to finally show itself in a validation report, I don't know.
And then I think Daniel wants to ask a question.
Thank you very much. I apologize, but for some reason, my chat is disabled to write something. So this is the only reason why I stand up my hands.
No, that's fine.
First of all, many thanks for the very enlightful and insightful talk. And I come from a small molecule world, and then 20 years ago, I turned as a structural biologist. What I miss is a relatively useful and nice interface between the PDB and the Cambridge Structural Database. There is any plan to make these two worlds jointly and speaking in order also in the field of a structure-based drug discovery where small molecules are, very common.
Well, so I'm now no longer in the leadership group of the PDB, but I talk to the people regularly. There have been various agreements between Cambridge and the PDB, formal agreements. So there are certain things that are allowed, but you know that the funding model for Cambridge
is different.
It's very different.
Different.
It's a problem. I can pass on your concern to the people at the PDB, as to what, do they have any plans? I know they would love to do it. And there was actually a period that was really great when CCDC's offices in America were next door to ours.
Yeah.
Same, but that was wonderful because you could say, why don't you do this or why don't you do that? But the funding model is the big problem.
Good. Yeah, this discussion I had many times with Edgar Meyer that I had honored to know during my PhD studies in Robert Hubert's lab in Martinsried. So he was there as a postdoc. So we had several discussions about this issue.
Doriana, I'm really, really glad you got to ask your question. I also, for some reason, have been thrown out of the chat, which is why I can't see any of the questions in the chat.
Well, I see the chat.
I see the chat, but I cannot write.
Oh. Any more questions? Can anyone
Somebody
see?
just...
So there's one more that's been posted into the chat and the Q&A. But it's, how can you extend the MMSIF to handle multi-scale structures up to organelles or cells?
In progress. Okay. So we have this project, if we could ever get it funded. This is not with the PDB, but it's the idea of having a cell SIF. So we have this is right now I live in California and I'm involved with people who are doing cell modeling, and Brenda is involved in this too. And they have come up with what they call cell SIF. But I don't know that it's been written up, but the idea is all there. Okay. So it's really, I think it's the natural next step.
That sounds awesome. We really want that. Sounds really, really good. Any other questions? If not, then there are still actually quite a lot of us still remaining. So thank you very much to everybody who's participated today. So big thanks to Helen, obviously. It was absolutely fantastic talk, really, really lovely to have that overview, and also to highlight the importance of collaboration, which I think is just so important, and lovely to be part of this sort of global community where we can all get to hang out together, even though it is online. So I will look forward, Helen, to seeing you in San Francisco at Biophysics. So if everyone could do a reaction, I'm going to do like a, I'm going to do an applause reaction. Thank you very much, everybody. Oh, John has put up his hand. John, have you, or are you doing clapping? John, do you want to say something, or is he? John might be clapping, I'm not sure. But okay.
He's got his hand up, I think.
Yeah, John, do you want to say something, or? Hello, John.
Oh,
We can't hear you.
Mute. It's muted.
Sorry, John, we can't hear you. Thank you, John. So, Helen, we will edit your talk and make it available online. And on behalf of the DRIIMB Consortium, we would like to thank you very, very, very much for giving a fantastic presentation. And everybody, there will be another DRIIMB talk. I think it will be the 24th of February. And it will be my friend Frank from Leeds talking about mass spec data. And Helen has already told us how important mass spec data is. So, oh, thank you. Yeah. So, yeah. So fantastic. That would be brilliant. Oh, and we've got Andrew, and then, oh, and Alexandra Bonvin. He's amazing as well from Haddock. Brilliant. Okay, well, a big thank you to everyone, and thanks everybody for all of your support with the technology today. We work together as a team, and so hopefully see many of you online again, hopefully in February, if not for subsequent DRIIMB talks. So thank you, everybody. Bye-bye.
Bye-bye.
Thank you,
Thank you,
Helen.
Helen.
Bye.
It was
Bye-bye.
amazing. Bye-bye, everybody.
Bye.
Bye, everyone. Bye. Bye.